So I guess I'll just sort of do a quick introduction, and then Anson's going to get underway with this work talking about using Nova floors in panel design on a conventional cytometer. And so I'm Sutton Thomas. I'm a scientist at Phytonics. We've been acquired by Thermo Fisher, so that's also going on in terms of expanding the antibodies that we'll have available. So that'll be really great. And then this is Anson Blanks, who is a customer success scientist at Phytonics, and again now part of Thermo Fisher. And he has done his PhD at VCU and in exercise physiology background. And the part that I can remember off the top of my head is the more fun part, because we try to make things interactive. So throw out your questions. Anson enjoys, craft beer, quiet rooms, and drinking craft beer in quiet rooms. So anyway, just throwing that out there as sort of a fun piece, but please throw your questions. I'll basically interrupt Anson at any point throughout the webinar, so I'm happy to do that.
Thank you, Sutton. Got all the fun parts in there. That's what's important. All right. So I want to thank everybody for joining us today. We'll go ahead and get started. So I'm going to be talking about flow cytometry panel design with Nova floors, and really how to maximize your cytometer to get the most out of your experiments. If I can get this to move. All right, there we go. So 3 pictures here, and they surprisingly do all have a common theme. So to start off, on the left side, so we've got Masaki Watanabe, who is the inventor of arthroscopic surgery. In the middle, we've got the Golden State Warriors, who are pretty recent 3-time NBA champions. And then over on the right, Larry David, who is the co-creator of Seinfeld, as well as Curb Your Enthusiasm. And what all of these individuals have in common is that they were game changers. So Dr. Watanabe invented a almost non-invasive form of surgery that's used worldwide. Golden State really leaned on the 3-pointer in the NBA and changed the game. And if you've ever seen Seinfeld or Curb Your Enthusiasm, you know that they are not your typical TV shows. So all of these people really changed how their fields operate. So in thinking about that, at Phytonics, we kind of asked, how can we change flow cytometry? And the answer to that question was by engineering a better panel. So we took engineered fluorescent labels with superior optical performance, which enables us to ask more questions per cell. And then, of course, we designed a panel for the highest data resolution. And that gives us the ability to enable a panel with more markers and higher resolution with no instrument changes. So just a quick overview. I'm going to show you how to take this 11-color T-cell panel, which I'll talk about more in depth a little bit later, and expand it out to a 13-color T-cell panel. And this is on a typical cytometer with no changes in instrumentation. So this is basically just panel design and using Nova floors. And expanding that 11-color panel to 13 is going to really allow us to dive deeper into the phenotyping, and in this case, see some chemokine receptors within these T-cell populations. So when thinking about dye performance, these are the major things that we have to think about, and how this dye performance actually impacts biological resolution and panel design. So number one, the first thing that we have to deal with is spectral overlap. So we know that dyes are going to have some spectral overlap, and usually we try and deal with that by compensation. 2 is going to be antigen density. So we want to follow the typical rules of flow cytometry and match lowly expressed or dim antigens to bright floors and vice versa. And then 3rd is going to be spillover spread. So this becomes more of a problem when we add more floors into a multicolor panel, and especially brighter floors. So even with compensation, there's going to be some spillover spread error, which is going to cause us to lose some resolution within our data. And number 4 is going to be the stain index, which is simply just going to be the separation between our positive and negative populations. I'm going to be talking about legacy dye performance. And when I say legacy dye, I'm just talking about your typical dyes that you probably all use multiple times. So PE tandems, all of your dyes that you've heard of. And with these legacy dyes, they typically have two major issues, with the first being cross-excitation. So cross-excitation just means that these fluors are going to be excited by more than one laser, an inappropriate laser. So if we look down here, especially at PE and PE dazzle, we know that they're going to be excited by the yellow-green laser, but they're also going to have pretty strong excitation by our blue laser. And an example of this over on the left side is going to be, this is PE dazzle on our Tune NXT, and we can see that this is just a single stain looking at BL2 off of the blue laser and YL2 off of the yellow laser, and we've got pretty strong excitation off of both of those lasers. So that's going to be something that we have to deal with. And then the second type of performance is going to be a broad emission, essentially. So with a ideal fluor, we would have a pretty narrow emission, so there wouldn't be much spillover, but we can see that that's definitely not always the case. So if we look at BV515, we can see that its spectra spreads pretty far across the spectrum. And because of that broad emission pattern, we're going to end up with a lot of spillover spreading error. So when we think about spillover spread, we like to think in an ideal world that we can deal with this with compensation. So if we take APC size 7 and PE size 7 and compensate them, this is what we would want it to look like. However, in reality, it's more of a perpetrator and victim relationship. So we can see that APC size 7 is spilling over or victimizing PE size 7. So it's spilling over into the PE size 7 channel where it's not supposed to be. And that's going to cause us to lose data resolution and maybe miss some of the small changes that we should be able to see. So when we think about spillover spread, again, that's going to impact our data resolution. So it's something that we have to take into account for, and we want to minimize as much as possible. And before we can do that, we actually have to define it and measure it. And one of the ways that we do that is by looking at the spillover spread matrix. So whenever you perform compensation in FlowJo, it will give you this spillover spread matrix. And basically, the higher the number, the more spillover spread, the more of a problem that fluor is. So we have a 4-laser Attune NXT with 14 detectors, which we would think would give us the ability to analyze 14 colors. However, we know that is not the case. Down here, we have the recommended fluorophores. Whenever we are designing a panel, especially in FluoroFinder, and we put, let's say, PE Dazzle and PE Sci 5.5 off of our yellow laser, we'll end up with this error message right here that pops up. And it's basically telling us that because we've put these fluors into our yellow laser and they're cross-excited by the blue laser, we can no longer use these blue detectors. So we lose BL2 and BL3. And stepping back just a little bit, whenever we're using these violet fluors, there tends to be a lot of spread. So whenever we add these specific blue BB510 and BB605, we're usually not going to be able to use this VL4 detector. So already our 14 detector tune is down to 11 useful channels. If we switch these fluors out, so switch the yellow over to the blue, still got the same cross excitation occurring, so we lose these 2 yellow channels. So no matter what we do, we basically only got 11 detectors that we can actually use. So taking those 11 detectors, we designed a memory T-cell panel using our legacy fluors and some common T-cell antigens. And these are the results from our panel. So we were able to pull out double negative T cells, CD8 positive, as well as our CD4 populations. And we also were able to look at NK cells and NK cell subsets. So overall, a pretty decent panel. So you're probably asking yourself, if I can take 11 colors and produce that panel, why do I need to change? And one of the reasons is going to be spillover spread or data resolution. So if we look at our legacy panel, over on the left side, we can see that there's this fishtailing effect, which is going to impact where we actually set our gate and where we're able to look at our positive versus negative populations. Versus if we drop in a NovaFluor, which I'll go into more detail about this in just a minute, we have a much cleaner population. It's going to be easier to set our gate and we can gain some resolution. So I've talked about resolution, and now I'll hopefully demonstrate the importance of resolution. So when we're talking about resolution, we can see this picture here that has a 20% resolution loss. You can probably tell what the picture is. It looks like bagels to me. Although if we actually look at the full resolution, we can see that there are some bagels, but there are also some puppies that are just trying to take a nap, and I don't think we would want to eat those. If we lose even more resolution, so let's say we add more floors into our panel, we're probably going to lose some resolution. The picture becomes even harder to see. So if we look at what's on the screen right now, you can probably say that there's maybe some blueberry muffins or chocolate chip muffins. However, when we look at the full picture, we can see that there are, again, some dogs that are looking back at us, not terribly appetizing. So resolution is very important. So given all of these effects of these legacy floors, what if you could design your own fluorescent label? So what impact would that have on your panel design and results? So you would want your label to meet 3 major requirements. You want it to be bright and clean with as little cross excitation as possible, consistent and stable, and just to throw it in there for fun, it might help, controllable brightness. So if you could control your floor's brightness based on antigen density, how would that impact your actual label or your panel design? And at Photonics, we have taken all of those 3 requirements and designed our NovaFluor labels. So NovaFluors are built on our Phyton structure, which is a DNA nanostructure platform. And within the DNA, we embedded fluorophores, which allows us to control the distance between the floors and really control the excitation and emission spectra. And it also gives us the ability to tune brightness. So we can basically take the same fluor emitting in the same, or being excited by the same laser and emitting in pretty much the same place, and tune the brightness based on panel needs. All of our spectra can be found on FPBase as well as the Photonics.com website and the Thermo Fisher Spectral Viewer. These are just our first 6 labels. We have recently released 13 new labels. So all in all, we've got 19 labels, and there are more on the way. So just a kind of a quick overview of how you can use these NovaFluors in your panel. They're really simple, and you can just drop them in and replace some of the legacy floors that you already have. So everybody's probably using FitC or Alexa Fluor 488. You can simply replace that with Nova Blue 530. Same thing with our Nova Yellows. So if you have PE or PE Dazzle, you can replace them with our Nova Yellow 570 or Nova Yellow 610. And the good thing is you won't have the cross excitation that you have with PE and the PE tandems. And that cross excitation is one of the things that a lot of Fortessa users kind of think that they don't have. So they look at their blue laser and they think, well, I don't have the problem with the PE and PE tandem cross excitation. That's not true. The cross excitation is still occurring. It's just that the way most Fortessas are set up, they have the trigon detector. So it's basically just engineering around the cross excitation of PE. It's not really eliminating the problem. So just a quick reminder of our legacy panel. You can see that here. And the spillover spread matrix that occurs with our legacy panel. And over on the right side, this is just going to be the sum of row sums, which is going to give us an indication of the spillover spread from the panel as a whole. And then we've got the spillover spread that's coming from individual floors. So using this spillover spread, that gives us a good idea of which floors we kind of want to attack and can replace in order to improve the resolution of our panel. So PE dazzle was one of the biggest contributors to spread. So in taking that in also with the fact that PE dazzle is cross-excitated and it's going to make us lose our blue detector, we simply just took PE dazzle out of the panel and replaced it with Nova Yellow 610. So same detector, same antigen, we're just pulling PE dazzle out and putting Nova Yellow 610 in. And that's going to allow us to open up this BL2 detector because we no longer have that cross excitation. So we've already gained a detector that we didn't have before. If we look at the spillover spread or the spillover spread matrix after we replace PE dazzle, we can see that there's a 19% reduction in that individual detector. So a 19% reduction for Nova Yellow 610 and a 4% reduction in the panel overall. And 4% may not sound like a whole lot, but if you think back to those pictures, any improvement in resolution can really impact the results that you're seeing. So when we look at the results of this Nova Yellow 610 replacement, replacing PE dazzle, you can see we pretty much get the same thing. So we can pull out all of our populations. So we still got our double negative, CD8 positive and CD4 populations. And I do want to highlight that if you're looking at these, our legacy floors that we're going to be replacing are going to be in red versus our Nova floors, which are going to be in blue. So if we replace PE dazzle, we can replace that legacy floor with a Nova floor, and that allows us to improve our resolution, but without really losing any other aspects of our panel. So we can see that the Nova Yellow 610 is not quite as bright as the PE dazzle. However, we do have a bit lower background, so we're still able to really separate out our positive and negative populations, which is what's really important. So going back to our original spillover spread matrix with the legacy panel, we can see that our per-CP psi 5.5 is another pretty big contributor to spread. So what if we leave PE dazzle in and just replace the per-CP psi 5.5? In this case, we went a little more complicated. So we kept, we removed per-CP psi 5.5 and replaced it with Nova Yellow 610. That does require us to move detectors, but we kept it on the same antigen. So we're just kind of moving the panel around a little bit. And if we take a look at the spillover spread matrix, we actually do have an increase in spread. So we do have some resolution loss, and that's really due to stacking of that yellow detector, and also our Nova Yellow 660 is a pretty bright floor, and adding the more or brighter floors in usually increases spread. But we end up gaining a detector because we no longer have the cross excitation to begin with. And if we look at the results of our panel, we're getting pretty similar results. So again, we can pull out all of our T cell populations that we had before. We're still able to separate all of our populations just like we were with the legacy panel. In addition to gaining a detector, we were also able to eliminate a tandem dye. So we can get rid of per-CP psi 5.5, which is a known tandem dye. And with tandem dyes, there's quite a few reasons why we would want to get rid of those. And number one is going to be that they're pretty notoriously unstable in solution. And that instability can really lead to some false positives, as we see over here on the right side. In addition, tandems are known to decouple differently on different phenotypes. So over here, we've got an example of decoupling on monocytes versus lymphocytes. Tandems break down very quickly in fixative solution. So this is freshly stained APC size 7 or cells with APC size 7. And then after 72 hours, we can see that there's a large increase in the positive signal, and that's due to the breakdown of that tandem dye into the individual tandem components. So it's breaking down into APC and into size 7. And then probably one of the most annoying, if you're trying to do a longitudinal or repeat experiments, is that tandems have a lot of lot-to-lot variation. So these are the same tandem dye, So it's the same floor on the same antigen, just from different lots. And you can see how much variation there is between these individual lots. So going back to the panel, we've replaced P-Dazzle, we've replaced Percy-P Psi 5.5. What if we replace both of them at the same time, which I think is the next logical step? That's going to allow us to open up these blue detectors. So now we have BL2 and BL3 that are open. And now we can add some more floors. So we can take this 11 color panel and build it out to a full 13. So in this case, we're going to add in Nova Blue 610 on CXCR4 and Nova Blue 660 to look at CD28. So you can, of course, choose whatever antigen that is of interest to you. These are just the ones that we kind of thought about looking at. So we've got our 11 color panel that we've now built out to our 13 color panel. And one of the things to notice, and I'll talk about it more in just a second, is going to be that these are Nova Blue 610 70S and Nova Blue 660 120S, which are going to be our brighter versions of these floors. And we chose this brighter version because these chemokine receptors tend to be more lowly expressed or not as densely expressed. So we want a brighter floor, and that's really nice because that allows us to kind of drop in these floors or drop in these Nova floors without completely redesigning our panel. So going along with that, so we do have two versions of our Nova Blue 610 and Nova Blue 660. So we have our 30S and 70S for 610 and 40S and 120S for 660. So you can think about the S designation as being like the wattage on a light bulb. So the higher the number, the brighter the floor. So our 30S is going to be brighter, or our 70S is going to be about twice as bright as our 30S, and our 120S is going to be about 3 times brighter than the 40S. And we can see an example of that here. So it's going to emit in the same detector and of course be excited off of the same laser, but it's going to be brighter. And this is really helpful when you think about panel design. So if you've ever done a theoretical panel design, and then when you actually run it, you know that, or you find out that your floor isn't bright enough for your antigen, this allows you to basically just get a brighter floor without completely redesigning your panel. All right, so going back to our 13 color panel, so now we've built it out. We've gone from 11 colors to 13 colors, and here are the results. So you're probably thinking, well, this is significantly less data than you were showing before, but that's only because we now have so much extra data, we couldn't get it all onto one slide. So we're able to pull out all of our T cell population. So again, our CD8 positive, double negative, and CD4 positive T cells. But now we're really able to dive deeper into the phenotype and look at the expression of chemokine receptors on all of our T cell subsets. So we've really got a lot more data here versus our 11 color panel. So if we take this 11 color panel, expand it out to 13 colors, we've increased our number of colors. What about our resolution? So this is going to be our spillover spread matrix using 4 Nova floors. And the next question becomes, okay, we've got 13 colors. Can we make that 13 color panel even better? And the answer to that, of course, is going to be yes. And we're simply just going to replace more of these legacy floors with Nova floors. So we're going to take out one of our big contributors to spread, PE, replace it with Nova Yellow 570, and then we're going to replace Alexaflor 488 with Nova Blue 530. So Alexaflor 488, not a huge contributor to spread, but replacing it with the Nova Blue 530 will help improve our panel, as we can see here. So when we take out those 2 additional floors and add in our Nova floors, we have about a 26% reduction in the overall spillover spread matrix, which is going to increase our resolution by about 26%, which again, if we think about the puppies and the muffins, that's a pretty big increase in resolution. So again, going back to our 13 color panel, we get very similar results as we had with the 4 novafluors, and we can really dive into the deeper phenotyping versus the 11. And if we look at the spread or the improvements in spread, we can see that we don't have nearly as much fishtailing, which is really gonna allow us to set those gates easier. And for, depending on what you're looking at, it's probably gonna improve your resolution to the point where you can maybe see some small changes that you weren't able to see before. So overall, we want you to get what you paid for. So if you paid for a 14 detector Attune or a 18 detector for Tessa or a Cytoflex, you probably got some lonely detectors that are kind of sitting around not getting a lot of use. You paid for them, put them to work. So basically you can really get what you paid for by getting rid of those pesky cross-excited floors. And doing that is going to allow you to no longer have to stop your analysis after you look at, in this case, get down to the CD4 positive level. You can really dive down a little bit deeper and look at the deeper phenotype. And again, we looked at chemokine receptors, but you can look at whatever fluor or whatever antigen is of interest to you.
Okay, Anson, we have a few questions right now. So the first one is about 40S versus 120S in terms of the brightness. So is the 120S 3 times brighter in terms of MFI or in terms of the separation index? And they were talking about the negative, looking like there was some background there.
Yeah, so it's going to be based on the separation index.
Yeah, so that's how that was measured. And then how is the spread measured in the bar graphs?
It's using the spillover spread matrix that's generated by FlowJo.
Yeah, so, you know, basically taking it for each of the colors, basically you can see, you know, what is that there, the row sum, and then you have the sum of the row sums, right?
Yes.
Right.
All right. So I've talked about bright and clean. I've talked about controllable brightness. So now I can show you some data on consistency, or show you some data on consistency and stability. So novafluors are very, very stable. So we can see an example of that here. So these are, again, our first 6 wave 1 novafluors. And this is at 4 degrees C on the blue line versus room temperature, which is going to be that orangish yellow line. We can see there's not really much of a difference between 4 degrees C and room temperature. So even if you have a forgetful graduate student or lab tech that leaves the novafluors out, it shouldn't make too much of a difference in their performance. novafluors also very stable in fixative. So I think this was 1% paraformaldehyde versus a fresh stain. And we can see that there's no difference in MFI or background even after 2 weeks of being in fixative.
Okay, Anson, I'm going to go ahead and interrupt you now since we're talking about fixative. We have a number of questions that have popped up, but one of those is, you know, how do these dyes perform with fixatives and permeabilization reagents?
So with all of the fixatives that we've tested, there's been basically no change. So they're really stable in fixative. So we've tested paraformaldehyde as well as I'm blanking on our other
1% formalin.
Formalin, yes, yep. Formalin.
And then go ahead.
Yeah.
And then we've also fixed with the intracellular, you know, step in the kits. So
yes. And what was the second part to that question?
About permeabilization agents.
Yeah, so we have, so we're going to be in the process very quickly of using methanol fixation. We haven't tested that yet, but we have permeabilized with the FOXP3 kit. And what else have we tested for permeabilization?
Yeah, so we also use the Thermo Fisher intracellular cytokine kit, that permeabilization. So one being basically cytoplasmic, the other one being nuclear. So we do have additional blocking coordinates that we recommend when you go intracellular for those. Methanol may be a different issue because we obviously can't pre-block in the same way in the intracellular. So that's still work in progress. So we've got, okay. and I might as well just throw a few more questions your way. Are these tested in PBMCs or ultracomp beads?
So the data that I've shown you has been PBMCs, but we have tested in ultracomp beads also in very bright staining.
Yeah, so you have the ultracomp and ultracomp plus. All right. Then how does DNase affect the antibody?
So DNase at low concentrations, we have found doesn't have much of an effect as long as it gets washed out. Don't think we've really optimized, how much or how long with the DNase. But yeah, as long as there's a wash step, we found that it works well. Want to elaborate on that?
Yeah, so it generally doesn't affect the phyton. It can sometimes affect the linker. So generally, I would recommend, if you're using DNase, say in your prep, I would wash that out. And then once you've washed that out, in terms of getting the single cell resolution. So we've got a few more questions. Do NovaFluors work with VersaComp beads? I'm just not as familiar with them. I've just used the UltraComp and the UltraComp Plus. So I don't think there's any beads that they don't work with, but that's just, I just would have to look at if there's anything specific about those beads. And then, is there a need of special buffer when combining different NovaFluor dyes, as the case with Brilliant Violet or UV dyes? No, we recommend using the basically NovaBlock in terms of blocking just, there can be sometimes monocytes binding non-specifically to dyes, but basically that is still compatible with all the other buffers, and it doesn't work. It's not similar to the Brilliant Violet or UV dyes where you really need that. And then, the question about any difference in wash buffers. I don't think we've really seen anything with any specific wash buffers that is an issue. And we've definitely used different ones there. So a NovaBlock is not the same as an FC block. So there's a question about that. And so that is, you can actually combine it with FC block if you want. So one is just really affecting more of the monocytes, or even like PE tandems, like sometimes you'll have this non-specific binding in terms of the monocytes. So it basically reduces that background.
Yeah, and if you're doing any kind of monocyte staining, you would definitely want to also include FC block.
Right. And then good question about viability dyes alongside the NovaFluors. So you want to answer that, Anson?
So no DNA intercalating dyes. Other than that, so we recommend amine reactive dyes, but as long as they're not DNA intercalating, I think we found that the viability dyes are okay.
Yeah, so just to elaborate on that, what happens if you do use an intercalating dye, such as 7-AAD, PI, or DAPI? We actually have seen this in practice. What it will do is interfere with the fluorescence, so you can't use those with that. But if you use an amine reactive dye, like a lot of the Live Dead or Zombie or any of those different stains, that's okay. And then would you use NovaBlock only with cells or with beads as well? Yeah, generally, I don't usually use NovaBlock with the beads because you don't have that non-specific binding. You certainly could use it, but you don't need to. Two more questions, and I'll let you go on, Anson. It's always great to see this kind of excitement, though. Have you looked at any hematopoietic panels, especially interested in CD34 and CD38 resolution? So we have not done that at this point.
I don't think so.
Yeah. So yeah, I would certainly be interested. COVID has certainly affected sometimes the ability to, what we're able to look at in terms of samples. And then the last question, using a cytometer, what is the advantage of the type of cytometer and using these? Yeah, so I think I did a lot of the work on building out the 40 and 45 color panels. And what I would say is that the NovaFluors are, basically very clean in terms of, this basically cross excitation and some of the spread. So it does, basically help with your overall panel. So for instance, you know, if you're getting excitation from every laser, you've got a lot of spread happening. So if you're using these floors, it actually does still help with the spillover spread error that you could see, whether that's a spectral cytometer or otherwise. All right.
Is that it?
Yep.
Right. Hopefully we'll get some more. All right, so consistency and stability together. So this graph is a little bit funny because it looks like there's only 3 lines, but actually each one of these colors, so the blue, green, and red, has 2 lines on each graph. And if you look down at the bottom, that's probably the easiest place to see right over here for Nova Blue 530. This is going to be our lot-to-lot consistency or lot-to-lot variability. And we can see that the variability between this first lot and the second lot that's pretty much the only place that you can see variability. And even this variability is less than 5%. So our lot-to-lot consistency is very, very good. So with all of that together, we have now engineered a better panel. So we've taken our fluorescent labels with superior optical performance, which enables us to answer more questions per cell or ask more questions per cell. And we designed a panel for our highest data resolution, which allows us to have a panel with more markers and higher resolution with no instrument changes. And I think that's the key here. So you can take whatever cytometer that you have and really get the most out of it. So we have 19 Nova floors that are currently available. For our CD4 test kits, which if you're kind of thinking about testing out our Nova floors, we have 2 different versions. So we have our Wave 1, which is our 6 original Nova floors. So Nova Blue 530, 610 30S, and 660 40S, as well as Nova Yellow 57, Nova Yellow
570 jumped off the list is what it happened.
Yeah, I see that's missing, but Nova Yellow 570, 610, and 660. And we also have our Wave 2 CD4 kits, which are our 13 additional labels that currently doesn't include Nova Yellow 720 because we had some manufacturing issues that were outside of our control. But if you order one of those kits, we will get the Nova Yellow 720 to you at some point later.
We got more questions. So one of them is just one question reminding it what the S means. So that is indicating the separation index. So it gives you sort of a relative brightness. So you can get, you know, the 610 in either of the 2 brightnesses, and the same with the 660.
Yeah, and I should explain more about the naming convention. It's very straightforward. I'm sure everybody's kind of figured it out already. So Nova Floor, whatever color, so Nova Floor blue is going to be excited by the blue laser, yellow by the yellow laser, red by the red laser. And then the number is going to indicate the peak emission. And then for any of the floors that had the S designation, like Seddon was saying, that's going to indicate the separation index.
Yeah, so you've addressed one of the questions. So what are the wavelengths for the yellow dyes? So somehow the Nova Yellow 570 got missed, but basically this naming convention tells you, you know, okay, this is, you know, its peak emission. So that's what's shown there. And then, so there's a question, is Nova Blue 660S 40S or Nova Blue 660 60S? And so it is the 40S. So basically a separation of 40 as measured on, you know, when staining human basically PBMCs with CD4. So that's what that is. And then another question about the, you know, spectral. Yeah, so it really does. We can certainly, the 40 color data and the 45. So using these dyes on the Aurora does actually give the same sort of help in terms of your panel. So what happens if you look at the spread, you can see that, you know, you can use these together. So, you know, you basically reduce that spread. So, you know, any of the PE and PE tandems still have that background spread. So as you build up to bigger and bigger colors, that spread is more of an issue. Oh, the emission of the laser. Good. Okay, I see what you're asking. So someone was asking actually, where did we measure the laser? So this was using, in terms of the separation index, that data was all done on the Aurora. So that is a 561 nanometer laser. I don't think you showed any of Steve Perfetto's work on the Symphony, but we have also run those with a 532 nanometer laser. So we do have some data there. Yes, and then several people are interested in how they can get antibodies and whether these are available in the Thermo catalog. And you want to answer that, or you want me to?
Yeah, so we have our in-stock antibodies, which are on the Photonics website. Those are ones that you can order, and we can have them to you in a day or so. For Thermo, we were obviously recently purchased by Thermo Fisher, and that is going to allow us to expand our conjugated antibody catalog. But that is going to take a little while. So right now our catalog is limited. I can't tell you exactly when we're going to have that expansion. But in the meantime, you could, if you're not interested in any of our in-stock floors, you could purchase a conjugation kit, which is a pretty straightforward, simple way of conjugating whatever your antibody of choice is to a NovaFluor. And then if you want something on a larger scale, so usually 500 micrograms or more, we do have our custom conjugation program where you would essentially either let us buy an antibody for you or send us the antibody and we would conjugate it in-house.
Okay. Yeah, and so these will be, depending on what you're asking there, also these will be enabled on the Thermo Fisher catalog. I think it's either sometime at the end of March or the end of April in terms of being able to order directly. So it's just a matter of getting, all of that sort of lined up to go on live on the website. But certainly you can still reach out to your Thermo Fisher rep if you're interested in some of that. And those will be available in, I think, Janssen, you may have a slide about what antibodies we offer. And, we do have both mouse and human antibodies. So there you go.
Yeah. So these are our mouse and humans that we have pretty much ready to go. I guess I can move on to this slide. So any color, any NovaFluor. So if you want one of these antibodies, you can get them on any of our available NovaFluors. We've actually just upgraded our website, so it's super easy. Just use a drop-down menu, pick your antibody, pick your NovaFluor color, and we can get that to you very quickly. So kind of the way these work is we have this oligo tag on the antibodies, which allows us to rapidly attach the NovaFluor to get that out to you very quickly. So you don't need to order a specific color or to meet a timeline. You can get any NovaFluor, any antibody. Okay.
I got two more questions, or maybe three. So one of these are the antibodies dispatched from the US only. So currently, Photonics is shipping basically worldwide, and we've had pretty good success in terms of shipping to Europe pretty quickly. So I think the only issues we've had is every once in a while there's a sort of COVID hold where there's a little more inspection there. So that and then I think Thermo Fisher will obviously longer term enable that through their channels. So that's just figuring out what the logistics of that are. And then a question about the conjugation kit. So do you want to discuss the conjugation kit that we have available?
Yeah, so we have our conjugation kit that is available for, that's available for our NovaFluors. I don't have the protocol in here, but it is a overnight process right now. So you're basically tagging your antibody with the oligo and then adding the NovaFluor onto the antibody. Pretty much the only major difference between the conjugation kit and our in-house conjugation kit is when we conjugate in-house, we do remove excess fluor, which the conjugation kit would not have that capability. But yeah, it's pretty easy to use. I should have the protocol in here.
Yeah. So yes. So you obviously you'd want to use our conjugation kit in terms of that, just because of there's the last step where you have to have the right DNA tag on there. And then do we have test sample sizes?
So we have smaller sample sizes. So for human, we have 25 test sizes, and for mouse, we have 25 microgram sizes. If you really just want to do a true test size of the NovaFloors, I would recommend the CD4 kit for kits.
Right. Okay. So.
And those are 10 tests for each 4.
Right. So then in terms of do we observe any non-specific binding with NovaFloors on negative 1 comp or ultra comp beads? I've done a lot of that work, so we really see very little background there. So that has not been an issue at all. And then are these dyes more additional options to the eBio portfolio, or some overlapping as well? Yeah, so it's one of the things we're super excited about. I mean, we really want to have, NovaFloors on more antibodies. So that's currently happening right now. So, if you have a favorite antibody that you're interested in, let us know. We're figuring out those kinds of things now. And yeah, so certainly that portfolio is open. So that's exciting. And then is the CD4 test kit available outside the US, Europe, to order through Invitrogen? So yeah, so right now the CD4 test kit is available worldwide. We've, basically as Phytonics, we've shipped everywhere. Thermo Fisher is in process of enabling this through their system. So, if you want to reach out to your Thermo Fisher rep or reach out to the smaller Phytonics team, either one will work. It's just not quite enabled on the website yet. As I said, that'll be March or April, depending on kind of what's happening there. So, but yes, you could get it, whichever way you want. And right now, we will actually be shipping those and can ship those pretty quickly.
Yeah, and for shipping domestically, we overnight, so pretty much anywhere in the US, we can get it to you the next day. For Europe, usually takes us about 2 days, as long as we don't get held up in customs due to COVID usually. And yeah, everywhere else, it's pretty much 2 days, 2 to 3 days, depending on where you are. But most places we've shipped about 2 days to get there.
Yeah. And some of that's just related to the time zone change too. Obviously, by the time we're shipping out, it's already the next day in Europe sometimes. So that's just a logistics issue. Is there a possibility of conjugating NovaFloors to something other than antibodies, cell binding proteins? Do you want to address it, or you want me to?
Well, I can start off the answer. I would say yes, there is the possibility. I don't know that we've actually tested any of that out at this moment.
Yeah, so the conjugation kit is, works through an amine reactive chemistry. So, basically, as long as that's not in the active site and affecting it, what you're looking at specifically, that shouldn't be a problem. So you could use the conjugation kit that's set up for antibodies, but there's no reason that it couldn't be set up for other proteins as long as they're not, I guess, too small or something like that in terms of being able to separate that out. But that would be the only issue there.
All right. Go back 1 second. So we are moving into the UV and violet lines. We are currently working on those. So keep a lookout. We will have some UV and violet NovaFloors coming soon. I can get my computer to move. All right, well, that was the end anyway. Yeah, big thank you to the Phytonics team. They really are superheroes. I think there's a lot of cool stuff going on. And with the Thermo Fisher acquisition, I think there's going to be even more cool stuff going on. We are definitely going to expand the conjugated antibody catalog. If you have any questions or want to make an order or anything else, definitely feel free to email me. So anson.blanks@Phytonics. You can also reach out to Seddon. Her email is seddon.thomas@Phytonics. Or if you can't remember that, you can always email info@phytonics.com, and that will get to one of us.
Thanks so much for all the great questions.
Yeah, keep them coming. We've got more time.
So.
Is there another question?
No, that's just a thank you. So that's great. Yeah, so I was trying to think of anything else that would be interesting for folks. But yeah, I think I'm just really excited about the new antibodies. I mean, that's certainly been the thing that I think everyone's wanted. So being able to get that expanded will be fantastic. And yeah, there's a lot of, new biology to come. And so it's an exciting time to be part of the overall team.
All right. Any more questions?
I don't think so.
All right, so I think that'll do it for us. Definitely want to thank everybody for coming.
Oh, I did have one question. Are data plots or slides available on our website?
So I know the 40 color data is available. There are kind of random plots available. I don't think we have more data available, like a big cache of data other than the 40 color.
Right.
Is that correct?
But I think if you wanted to look at, for instance, this webinar, it has been posted on the Phytonics YouTube channel, and I think Thermo Fisher is also going to start posting recordings, so that should be available to you to be able to watch. The slides themselves aren't available at this point, but if you want to rewatch the webinars, those are definitely available.
All right. Any more questions? Yeah, so if you're using a conventional cytometer, expand your panel, get more biology. You will have to clear out your schedule a little bit so you can do the analysis. And if you're using a spectral cytometer, clean up your panel, improve the resolution, really see what's going on.
All right. Thanks, Anson. Thanks
All
to
right.
everyone for coming.
Yeah, thank you.